MCAT 2017 Cram Biochemical and Biological Foundations of Living Systems. Passage 2, Measuring Glomerular Filtration Rate of the Kidneys with Inulin. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections to answer the questions that follow. This passage is really manageable, and so are the questions, so good luck and happy listening, or happy reading rather. Paragraph 1. In chronic kidney disease, the kidneys lose their ability to effectively filter waste products in the blood because of damage to the glomeruli of nephrons. Kidney function is determined by measuring glomerular filtration rate, or GFR for short, the volume of plasma that the kidneys filter through the glomeruli per unit time. The gold standard for measuring GFR is through the use of inulin, not insulin. I know a lot of people probably automatically saw insulin. This is inulin, a carbohydrate produced by many plants, such as onions and garlic. Inulin is not endogenous in humans. A specified mass must be injected into a person's bloodstream in order to measure GFR. Paragraph 2. Inulin is useful as an indicator of GFR because the kidneys handle it in a unique way. Unlike most other substances in the blood, inulin is neither reabsorbed into the bloodstream after filtration nor secreted through peritubular capillaries. Thus, the amount of inulin cleared through the urine is indicative of the amount of plasma filtered by the body's glomeruli. GFR can be calculated using equation 1. Equation 1. Urine concentration refers to the concentration of inulin in a sample of urine. Urine flow refers to the amount of urine produced in a given period of time. And plasma concentration refers to the concentration of inulin in blood plasma after intravenous injection. Table 1. Group A, control, received no treatment. Group B received 20 milliliters of intravenous saline solution. Group C received 20 milliliters of intravenous saline solution with 1 milligram of furosemide. Paragraph 3. Researchers investigated how GFR was affected by furosemide, a diuretic medication that prevents sodium reabsorption in the loop of Henle. To do so, they observed the clearance rate of inulin in 3 groups of healthy cats that received different treatments. Their results are shown in Table 1. So Group A is the control group. Nothing was given to them. Group B is the control group. They were just given saline solution. And Group C was treated with the medication. You can see here how their inulin concentration in the urine is significantly lower than the other 2. Okay. Bumetanide is a diuretic drug that prevents reabsorption of salts and other solutes from the ascending limb of the nephron. How would bumetanide affect the body's rate of inulin clearance? Would it A, it would increase inulin clearance by preventing reuptake by the kidney. B, it would decrease inulin clearance by decreasing renal blood flow. Or C, it would have no effect on inulin clearance. I'll give you a moment to think. If you need to, press pause and go back to view the reading of the passage to search for the text support to answer this question. All right. Okay, so this question is really not that bad. Some people may think you need outside knowledge or an in-depth knowledge of kidney clearance in order to figure this out, but actually all the information you need is contained in the passage. Okay, so inulin is filtered through the glomeruli. Okay, so here goes the inulin being filtered through the glomeruli, but not reabsorbed or secreted. This makes inulin clearance proportional to the glomerular filtration rate, or GFR. So how would, oh gosh, I have a terrible time with this word, bumetanide affect GFR? Well, notice that bumetanide has the same mechanism of action as furosemide, the drug that was used in the study in the passage. And how did furosemide affect GFR? Well, to know how furosemide affected GFR, we would have to compare group B and group C, because remember, group A is the control. And without actually having to perform the glomerular filtration rate calculation, we can determine the relationship between GFR for both groups B and for group C. We can compare these two, okay? So one is just with salt and one is with salt plus the drug, okay? So notice that group B has double the urine concentration of inulin and half the output, which, according to equation 1, if we were to just, imagine plugging these values in, would make GFR equal for the two groups. Because if the urine concentration is double, 500, and the urine flow is half, 12 milliliters versus 24 milliliters, then it's going to be essentially the same, okay? So 500 times 12 is going to give you 6,000 in the numerator, and 250 times 24 milliliters of output is also going to give you 6,000 in the numerator. Notice all the denominator values are the same, so the overall GFR is going to be the same, okay? From this, we can infer that butanamide will have no effect on GFR. So there's no effect on inulin clearance, and the correct answer choice is answer choice C. In the nephron, blood flows from the afferent arteriole to the glomerulus, or the glomerular capillaries. I have a difficult time with that one. To the efferent arteriole and to the peritubular capillaries. The efferent arteriole, this region right here, most closely resembles which of the following vascular structures? Is it A, a fenestrated capillary? Now let's just do a quick review of what each of these structures are, okay, each answer choice. Fenestrated capillaries have pores in the endothelial cells, as seen here. The cells that line the blood vessel. They're usually 60 to 80 nanometers in diameter. That's how you distinguish them from other capillary types. And these pores are spanned by a diaphragm of radially oriented fibrils and allows small molecules in limited amounts of protein to diffuse. Okay, so this is a quick overview of a fenestrated capillary. Or is it B, a thoroughfare channel, as pictured right here? So they're also known as meta-arterioles, and they exist in mesenteric microcirculation, and the name was originally conceived only to define thoroughfare channels between arterioles and venules, but it's expanded since, okay? Or is it C, sinusoidal capillaries? Sinusoidal capillaries are a little bit different from fenestrated capillaries, which some people might confuse the two. A sinusoid is a small blood vessel that is a type of capillary. It has its endothelium is similar to fenestrated endothelium, but the pores are much larger and sometimes continuous. Sinusoids are actually classified as a type of open pore capillary. I said continuous or discontinuous, as opposed to, continuous or fenestrated types, okay? So this is just a really quick overview. If you want to do in-depth study on this, go ahead, but I recommend doing more questions than you do in-depth review. I know that seems antithetical, but trust me, it works. Or D, hepatic blood vessels. This is the hepatic blood vessel, this blue thing pictured here. So they're the most notary type in the human body would be the portal vein or the hepatic portal vein. And it's a blood vessel that carries blood from the GI tract, the intestines, to the spleen, and the liver. All right, so this is a little bit of anatomy for you. This deck of cards is really good because it covers a lot of good anatomy, okay? So I'll give you a moment to think. All right. Okay, so the glomerulus is a cluster of capillaries. Typically, there's only one bed of capillaries between two larger blood vessels, but with the vascular system in the nephron, there's like blood vessels between two capillary beds, one pictured here and one that you can't see beyond the peritubular capillary, okay? So basically, when a capillary bed recollects into a larger vessel, earlier I showed you the hepatic portal system. Here we have the capillary beds from the small intestine recollecting into the hepatic inferior mesenteric vein and then larger hepatic portal veins. Okay. This is also another example of a capillary system, although I could have shown you the heart. I prefer this one. So, yeah. Basically, again, when a capillary bed recollects into a larger vessel and then divides into capillary beds again before returning to the heart, you can see the capillary bed here returning to the inferior vena cava, which will go back to the heart. This is called a portal system. And the afferent arteriole connects between two capillary beds, the glomerulus seen here, and then another one beyond this point, which I don't know the name of right now. I can't recall. Therefore, it resembles a portal blood vessel. Okay. All right. So that's it. In which order would inulin travel through the following renal structures? 1. The collecting duct. 2. The proximal tubule. 3. The afferent arteriole. 4. The efferent arteriole. 5. The glomerulus. I can't pronounce this. 6. The distal tubule. And 7. The loop of Henle. So is it order A, order B, order C, or order D? Definitely press pause if you need to, and I'll give you a moment to trace the path. This is a really good renal anatomy question. It's really good. These anatomy questions do come up, although I think the biochem aspects are a little bit more prevalent. All right. Okay, so inulin is neither reabsorbed nor secreted, as mentioned in the passage. So once it's filtered, it will follow the path of filtrate, then proceed to urine. Okay? So blood first approaches the glomerulus through the afferent arteriole. And filtrate flows from the glomerulus, I don't know how to pronounce this, to the proximal tubule. So yeah, I think like this is like the proximal tubule or something. And then it's going to go to the loop of Henle. Okay, the filtrate will proceed back up to the top of the loop of Henle into the distal tubule. So this must be the distal tubule region over here. And then it goes to the collecting duct. So basically, I'm trying to say is that it's going to flow from the afferent arteriole to the glomerulus, 5, to the proximal tubule, as I mentioned before, to the loop of Henle, then to the efferent arteriole, and last but not least, the collecting duct. So the correct answer choice is going to be answer choice A. All right. Based on the measurements in Table 1, which substances or substance could be used to increase GFR? Is it A, inulin, B, either intravenous saline or furosemide? Remember that both groups B and C were treated with intravenous saline, but group C received furosemide. Is it C, intravenous saline, or is it C, furosemide? I'll give you a moment to think. All right. Okay, so based on the measurements in Table 1, what we can do is we can use equation 1 as well to calculate the GFR, the glomerular filtration rate. Okay, and we can do this for each group, but taking a long time to do each calculation would actually not be efficient. So instead, we can try to use the basic properties of the equation to see the relationships between the GFRs of the three groups, okay? So since the starting plasma concentration, which goes in the denominator, is the same for all three groups, we can ignore this data. Now we just need to multiply the urine concentration times the urine flow rate. Okay, So the first thing we can do is compare group A to group B. Notice that the urine concentration is cut in half, while urine output quadruples. So taken together, this means that the GFR doubled when the intravenous saline was administered. Okay, so here we have like 1,000 micrometers. Micrometer milliliters, I'm not even sure if that's a factor. And here we have 6,000 of these same units, okay? The only thing left to do is determine whether group C, which also received furosemide, also increased the GFR, which we can do by comparing the GFRs of both groups B and C, or we can, because we know it increased here, but will it increase from here to here as well? The urine concentration of group C is also cut in half, while the urine output doubles from group B to C. So taken together, this means that there was no change in the GFR. So we can conclude that furosemide does not have an effect on the GFR, okay? And so the only thing that would ultimately increase urine concentration as well as increase urine flow and overall increasing the GFR would be intravenous saline. So the correct answer is answer choice C. The researchers would also like to measure the effects of diuretics on total renal plasma flow. To approximate renal plasma flow based on a urine sample, they would want to measure the urine concentration of a substance that is treated by the kidneys in which way? A. Filtered and reabsorbed. B. Filtered and secreted. C. Reabsorbed and secreted. Or D. Filtered only. I'll give you a moment to think. Definitely refer back to the passage if you need to by opening up a second window. All right. Okay, some people mistakenly chose filtered only because they referred to the highlighted portion of text that says inulin is neither reabsorbed into the blood after filtration nor through the nor secreted through the peritubular capillaries. Thus, the amount of inulin cleared through the urine is indicative of the amount of plasma filtered by the body's glomeruli, okay? But this is the glomerular filtration rate. Here we're looking for renal plasma flow, okay? So this is a different measure. To approximate renal plasma flow, the ideal substance for researchers would be one that is entirely deposited into the urine after exactly one circulation through the kidneys, okay? So we're looking for something that's basically reabsorbed and secreted. When the glomeruli filter plasma, only about 20% goes through, while the rest stays in the blood. Therefore, a substance that is only filtered doesn't give us an accurate approximation of renal plasma flow. Reabsorption is when a substance is taken back into the blood from the filtrate, okay? So secretion is when a substance is transferred from the filtrate it's when a substance is transferred into the filtrate after the blood has already passed the glomerulus, okay? So to maximize the amount of a substance that is transferred to the urine, We would want a substance that is both filtered and secreted. So the correct answer choice is answer choice B.